over the course of this semester, we have talked about how your immune response and your immune system are composed of basically two parts. You have adaptive immunity and innate immunity. Adaptive immunity you kind of have to build up through exposure to pathogens, and eventually your body recognizes them and knows what to do. However, in innate immunity, you have a certain defense that just naturally occurs. You don't have to have already seen the pathogen, your body just kind of knows what to do. In this video, I'm going to focus on your first line defense in your innate immune response, which is complement. Now, complement is basically a pathway that is activated by plasma proteins made by the liver, and these proteins are found in blood, extracellular fluid, and lymph, and they are activated pretty much as soon as they see a pathogen that's crossed an endothelial barrier. Now, there's three arms of complement activation, and these pathways are the alternative pathway, the lectin pathway, and the classical pathway. So to kind of illustrate these, I drew this out right here. Now, your alternative pathway, this is the most common and is the first that acts naturally. This is when the pathogen surface kind of creates an environment in which complement can just naturally occur. And the second is the lectin pathway, and this involves mannose-binding lectin. Basically, that is when mannose-binding lectin binds to a pathogen surface and induces complement activation. And then you have the third to activate, which is the classical pathway, and this is when a C-reactive protein or an antibody binds to an antigen that is specific on the pathogen surface. Now, although these three pathways differ a little bit in how they occur and how they play out in the body, they do have one common goal, and that is complement activation. And complement activation during this process cleaves C3 into C3a and C3b, and C3b binds to the surface of a pathogen and induces death of a pathogen. There are different ways that this can occur. First, you have the recruitment of inflammatory cells. The second is opsonization of pathogens. Now, this is very common. This is when C3b binds to a pathogen and kind of marks it for phagocytosis by a macrophage, which we will be talking about more in detail later. And lastly, you have perforation of a pathogen cell membrane. That can be caused by a membrane attack complex, which we will also talk about in just a few minutes. All three of those end results, however, do lead to the same goal, which is the death of the pathogen. So that is the main goal of complement. Like I mentioned, alternative pathway is the most common form, so we're going to look at that one in just a little bit more detail. I kind of tried my best to draw it out right here, so we'll see how that goes. First, you start with an antigen and then C3b, which is this red dot right here. Those two bind and form this little complex here in the middle. And then three factors, factor B, factor D, and properdin, bind to C3b and form what we call C3 convertase. Now, C3 convertase takes C3 and splits it into C3a and C3b. Now, C3a is the smaller of the peptides that is made from C3 convertase, and it goes off and is involved in inflammation. I will talk about that a little bit later because it is very important, but for now we're going to focus on C3b and what it does. So C3b can do two things. First, it can go back up here, bind to another antigen, and cause more C3 convertase to be made, or it can be used in opsonization, in which it binds to a pathogen and marks it for phagocytosis by a macrophage. Briefly, I'm going to tell you about the membrane attack complex because it kind of is made the same way. First, you have up here C3 convertase and then properdin. So down here you have those two. If you add more properdin to the C3 convertase, it forms C5 convertase. And C5 convertase does the same as C3, just with C5. So it splits it into C5a and C5b. Again, C5a is the smaller of the peptides that's produced here, so it goes off and is also involved in inflammation, which I will also talk about in just a minute. But C5b reacts with C6, 7, 8, and 9, and these form what we call the membrane attack complex, which basically opens up a hole in the membrane of the pathogen and causes the cell to lyse open, also killing the bacteria cells, so that's very important as well. Okay, like I mentioned there, C3b is what marks the pathogens for destruction by a macrophage. I'm going to try my best to illustrate how this happens. I tried to draw it out. I'm very sorry if it's not very good, but basically this is what happens. So on this side, this is the bacteria, and these right here, those little round things hanging off of it, are the C3b peptides that have bound to the pathogen. Opsonization has occurred here and has been marked for phagocytosis by this macrophage down here. This is a lysosome inside of it. It will be important later, but not right now. These right here on the surface of the macrophage are known as CR1, which is complement receptor 1. In the second box, you can see that C3b has bound to CR1, and then in the third box, endocytosis has started to occur. So it is taking the bacteria inside of the macrophage, and then in this fourth box, the membrane has fused, again, the macrophage membranes. It's no longer endocytosing it. It's already inside of the cell, and it has formed this vesicle, which we call a phagosome. Now, in this last box right here, the lysosome that was originally right here has fused with the phagosome and formed what we call a phagolysosome. It releases enzymes and other things into the phagosome that break down the pathogen and cause it to die. Sorry. Okay, so the last thing that I want to talk about is we're going to go back and talk about how C3a and C5a are used in inflammation. So, C3a and C5a can sometimes cause anaphylaxis to occur, which is why these are known as anaphylatoxins. I'm not sure if I'm saying that exactly correct, but I can show you how it's spelled. Basically, C3a and C5a cause vascular permeability to increase. So in two different forms, it can cause inflammation to occur, basically by releasing more things into the area of infection. So we're going to focus over here first. Basically, increased fluid leakage from the blood vessels can cause plasma proteins and complement proteins to be released to the site of infection. You can see them leaving the blood vessel right there. And then on this side, you can see that it can also cause increased migration of neutrophils and monocytes to the area of infection. And these have microbicidal activity, so they can help to kill off the pathogen as well. The more of these cells and the more of these plasma proteins that are released into an area of infection, the more inflamed that area gets, which is why they are involved in inflammation. So, I have gone over today the three main pathways of complement, talked in detail about the most common and how it works, and how the two products of that complement pathway can be used to either kill the pathogen through phagocytosis with a macrophage, and how it can lead to inflammation. So I hope that helped, guys, and good luck on finals.